pretty good. How are you guys feeling about exams? Let me know in the chat. How's the studying going? Have you been started studying? Perfect. Stressed? Nicola says she's stressed. Well, that's actually expected. I mean, what else can you feel? You had a mock exam today. Paper 3 was so ass. Yeah, paper 3. Oh, I really hate database questions too. Um, how well do you guys know your option topics? Is it too late if I don't understand and know anything? Um, I don't think it's too late at all. I You just spend time focusing on things that you have more difficulty understanding, so like the harder stuff you find. Um, and try working on those and perfecting those. And then over time you'll see an improvement. I think you have lots of time. You have like 20 days, I think. My bio teacher is yeah, my bio teacher wasn't that good either, but here we are. I haven't even studied my option topic. Yeah, I studied for my option topic. Um, I think like 2 days before, 3 days before. And I was so stressed, but we made it. Can we start? Yeah, we can. We'll start in like 2 minutes. Do you give additional classes? Yeah, we do give additional classes. I was just going on our website and checking those out. Mm, yeah, start it. Yes, if you start studying with your worst subject, that's a pretty good way to start. Are you a student or a teacher? I'm actually a graduate, so I did IB last year, and I teach now. You entered the mock without going through the option topic. Yeah, what option are you doing? Because I feel like for my option, I did option D when I took the exam. A lot of the content was already stuff that we learned previously during the course, so it wasn't too hard. But I know for like option A, yeah, so option D, it's the heart, I think, the liver, digestion, and stuff. Yeah, okay. So we'll go ahead and get started. It's 8:05. So let's do that. Hopefully you guys can see my screen and everything. But if something goes wrong, just let me know. And let me know and we'll get started, okay? Perfect. So paper 1, this is an SL paper. It is time zone 2. So for reference, if you want to go look at it again, we can share this worked out past paper with you later. But so it is 45 minutes. Okay. It's 45 minutes, and that means you have about 1.5 minutes per question. Okay. And that means that when you're in your exam and you're sitting and you're watching the clock, you should, at around 22 minutes, so 22 minutes, you should be at question 15. Okay, so use that as a basis when you're looking and you're sitting in your exam and you're looking at the clock. When it says minute 22 or 22 minutes left, question 15, I should be on question 15. That's all about, that's everything basically you need to know. And this actually 30 marks. Okay, and that's little compared to your paper 2, but I think it's still good ways, because in paper 1 there are some easy questions and hard questions, so try to maximize as many marks as you can get. I can't say I have access to the worked out answers. We can find a way to upload that. We'll work that out later. Okay, but perfect. Let's just go ahead and start with question 1. How would surface area, volume, and surface area to volume ratio of a cell change as the cell becomes bigger? So my cell is becoming bigger. And how does that change if the cell does not change shape? So I'm going to give you a couple of seconds, so like let's say 30 seconds to try to reflect on this question, and then I'll go ahead and explain it in about 30 seconds. Okay, so I'm seeing a D in the chat. Anyone have any other ideas? I'll give you a couple more seconds. Okay, we have another C, B. Okay. So the answer is actually B. And we'll go ahead and explain why. So if a cell is becoming bigger, so I like to draw it out. Sometimes it's very helpful to draw it out. So if you draw out, here's my cell, and over time it's getting bigger, 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 bigger. And my surface area is basically, when I think about it, I think about it as my cell membrane. So this is my cell membrane here, here, here. So as my cell is getting bigger, and it's saying the shape doesn't. Oh, hello, hopefully you guys can see me. The phone cut off, so my camera cut off. But can you guys hear me now? Is everything good? Okay, perfect. Okay, let's continue. So my cell is getting bigger, and it's telling me in the question that my cell does not change shape. So I know that it's just going to stay the same circle or the same, ideally the same shape as it was before. So I just drew it out here, and I can see that my cell is not changing shape, but it just keeps increasing. Okay, and my surface area, as I increase, and my cell gets bigger, I notice that my surface area or my cell membrane actually increases. So that's why I know my answer for surface area is going to be increase. Okay? And so I can already cross out D because surface area does not decrease. Okay? And then now I need to look at my volume. So my volume, as my cell is getting bigger, I notice that my volume also increases. So I know that for volume, it's also going to increase. So it's not going to be C. So I've already crossed out C and D, and that's a pretty good um, first step into answering my question. It's definitely not going to be C and D. Now let's look at the surface area to volume ratio. My surface area to volume ratio is essentially looking at the ratio between my surface area, this here, and my, I'll do it in red, volume. Okay? And I notice as my cell increases in size, my surface area is increasing, my volume is increasing, but actually my volume is increasing at a much higher rate. So if I just told you based on looking at this drawing, what is um, taking up more space? Is it going to be the red that I'm drawing, or is it going to be the green that I'm drawing? And it's actually going to be the green. So that's how the surface area to volume ratio works when you're looking at cells. Usually the surface area doesn't increase as much as the volume. So my surface area is going to increase at a much slower rate. So it's going to be surface area is going to be much smaller rate than my volume. So my surface area to volume ratio is going to decrease. Okay? So that's why our answer is B. Did that make sense for everyone in the chat? Hopefully, yes, yes. Okay, perfect. Perfect. Okay, I'm gonna move on to the next question. Which cell component is found in eukaryotic cells, but not in prokaryotic cells? I'm going to give you another 30 seconds to solve this one. Why? Okay, I'm going to give you 30 seconds to solve this one, and I'm going to go ahead and answer any questions you have in the chat. And we're on question 2, by the way. Wait, why does the surface area decrease? The surface area doesn't decrease, but it actually increases, but it increases at a rate smaller than that of the volume. So the volume increases much, much more than the surface area increases. So the surface area to volume ratio is going to actually decrease. Which year is this paper from? It's May 2023. Okay, I'm seeing a lot of D's in the chat and a C. Okay. Give you 10 more seconds and then go ahead and answer that. So surface area is lower than. Yes, exactly. Yes, Chris got it right. Okay, I'm going to go ahead and answer this question. So the way I like to think of it is when I look at eukaryotic cells, I think those are cells with a nucleus. Okay, and these are without. Okay, and that's how I like to think of it. And let's just go through the questions and the answers and see which one matches. Mitochondria for respiration. Actually, prokaryotic cells, if you know prokaryotic cells don't have mitochondria for respiration. So our answer is actually going to be A, but let's go through the other answers and eliminate why the answers are not B, C, or D. Okay, so DNA containing genetic information. Both eukaryotic and eukaryotic cells have DNA containing genetic information. I think what they're trying to trick you here is with the nucleus, but we know that eukaryotic cells have nucleus, prokaryotic cells don't have nucleus, but they do both have DNA. Okay, so it's not going to be this one because DNA is contained in both. Now, ribosomes. Ribosomes for protein synthesis are actually also in both prokaryotic and eukaryotic cells. So our answer is not going to be C. So this is in both, and then this one's also in both. Okay, and then a cell wall to maintain shape. I can see that's where a lot of people um, kind of fell for the trap, because when you think of eukaryotic cells, you usually think of animal cells, but remember, eukaryotic cells can also be plant cells. And plant cells do have cell walls. So that can lead me to conclude that both prokaryotic and eukaryotic cells have cell walls, so it can't be D. So through the process of elimination, I was able to figure out that it was A. And just to add more information, prokaryotic cells do not have mitochondria for respiration. And the way I like to remember that is prokaryotic cells don't have double membrane bound organelles. So that's why our answer is going to be A. How does prokaryotes not have mitochondria? They don't have mitochondria because they don't have double membrane bound organelles. So they don't have chloroplasts and mitochondria. It's one of the things that you should just know about prokaryotes. Do you have any other questions, or should I move on? Is this A levels? No, it's IB. Can I zoom out a bit? Yeah. Okay, I'm going to move on to the next question because I don't see any more questions. Okay, the drawings are from two original papers that are the structure of the cell membrane. So unfortunately, we don't have the drawing because of copyright reasons, but that's okay. But it's just telling me that it's the structure of the cell membrane. No problem. We actually don't need the drawing to answer the question, but we can go ahead. Okay. For what reason is the Singer-Nicholson model considered a better representation of the cell membrane than the Davison-Danieli model? So I'm taking this Singer-Nicholson model and comparing that to the DD model. Okay. And I'm just going to go ahead and draw out the different models while you guys give me some answers in the chat. Okay. So I'm getting B's. It's either B or C. It's either B or C. C, C. Organelles. A. Okay. That's pretty good. I'm getting a lot of C's, and that's actually correct. The answer is C. And let's go over why that is. Okay. So the Singer-Nicholson model actually represented the cell membrane like this, like in this image here. We have a phospholipid bilayer. So these are my phospholipids. It's not a very good drawing, but these are my phospholipid. And what actually differentiated his model of thinking compared to the Davison-Danieli is that he said that we have integral proteins. So proteins that go through our phospholipid bilayer. So you can see an example here, okay? And that is compared to the Davison-Danieli model when he said that there's no proteins that go through the membrane. We actually have a thick layer of proteins above and below, right here. So one suggested that proteins go through the membrane, and one said that proteins are just on the outside like this, okay? And so the question asks, what is considered a better representation of the cell membrane? Okay, let's go through the answer. It has extrinsic proteins, and that means that they have proteins on the outside. And the Davison-Danieli model actually both said that there's proteins on the outside. And if you look at the Singer-Nicholson model, there's also believed to be proteins on the outside. So this actually is seen in both. So it wouldn't be considered why it's a better representation. Okay, it shows how the phospholipid bilayer is arranged. Both models actually show how the phospholipid bilayer is arranged. So it wouldn't be this one either. Okay, it helps the understanding of the fluidity of the cell membrane. And the answer is actually C, and that is because in the Davison-Danieli model, he believed that these proteins on the outside were here, and these proteins would have meant that the flow of the membrane would have been very restricted. Because if you think of proteins, you think of them as solid and rigid structures, and they would have actually affected the fluidity of the membrane. While here, you only have proteins in the middle sometimes, and sometimes proteins on the outside, but no proteins at the top or at the bottom like this, preventing the fluidity of the membrane. And that's why our answer is C. And then for D, it says it shows how oxygen can diffuse in the cell. And this is kind of like a random option that the IB has given you because none of them really show how oxygen is diffused into the cell. None of the models show that. Okay, did that make sense for everyone? Hopefully, yes, 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 yes. Okay, let's move on to question 4. You guys over anything? You guys always go for elimination? I think process of elimination for me works the best. But if you have trouble doing elimination question, like the process of elimination, you can find other techniques. Okay, question 4 says, the diagram shows protein channels involved in the passive movement of a substance into the cell across the membrane. What describes this movement? So I have a passive movement, the keyword is passive, and I can see an image here of the passive movement. I'm going to give you guys 30 seconds to try to answer this. Okay, and then I'm going to go ahead and go over the answers. So when you're thinking of passive, think, does it need ATP or does it not need ATP? What else? Okay, I'm getting a lot of B's in the chat and a D. D, okay. D. B, D. Okay, let's go over it. So if you said D, you were actually correct. The answer is D. And we'll go over why D is the correct answer. So it's talking about passive movement. And straight away, when I think of passive movement, I think about no ATP, right? Okay, no ATP and from high concentration to low concentration. Okay, let's go over the answer options, which describe this movement. Energy of ATP is used to transport substances into the cell. Well, we already decided in the beginning that passive movement doesn't involve ATP. So this is actually for active transport, not passive. Okay, substances can move from areas of low to areas of high concentration. That is actually also incorrect. That's also for active transport. And actually, For passive, it's areas of high, so it's going to be high to low in passive movement. Okay, the proteins ensure the substances is only in one direction. So this is a question, a point that maybe your IB teacher didn't go over in class. But actually, what happens is in the general movement of stuff is that there's going to be movement of these molecules in both directions. So some of them are going this way, some of them are going that way. But the key thing to consider is that the net movement, so that's the key word that you have to use in your when you give a description. So net movement is actually in one direction. So movement is happening both ways, but the net movement is in from high to low. So that's why the answer is not C, because substances do not only move in one direction, they actually move in both directions. But again, the net movement is from high to low. Let's look at D. Net movement occurs until the concentration in and out of the cells are equal. And that is an answer that we like to see because we know that cells and just life in general likes to maintain equilibrium, and we work and work and work until we have equilibrium or things are equal. And in this case, it's the concentration is equal on the in and outside of the cell. Does that make sense for everyone? Oh, I read B as high and low. Yeah, that's a common mistake. I would suggest maybe underlining things so you can make sure you don't make those mistakes. My camera went away again. I apologize for that. Okay. I apologize for that. It keeps happening. This is not AP Bio, this is IB Bio, but yeah. Hopefully that went well for everyone. Okay, let's go on to question 5. The image shows a cell from the root tip of an onion, okay, during late prophase of mitosis. How many chromatids, so the key word in this question would be chromatids, are present in the cell? I'm going to give you 30 seconds to work this out, and also keep a look at this number here. So 2n equals 16. What does that mean, and how can we help use that in the question? Okay, I see a C in the chat. C. A, C. Give you a couple more seconds, and then I'm going to go ahead and explain it. Okay, perfect. I'm getting a lot of C's. That's really good. So the answer is actually C in this case, and I'm going to go ahead and explain why. So it's telling me that there are 2n equals 16 chromosomes during late prophase of mitosis. So it's telling me that there's 16 chromosomes during late prophase of mitosis, and I know that 16 is my diploid number because it says 2n. If it says just n, that would mean my haploid number. But again, it's just saying my diploid number, and that's telling me it's 16. Now, if we look closely, the question is actually asking me how many chromatids are present in the cell. And that's when I have to make sure I understand what a chromatid is versus what a chromosome is. So if I look here, this is a chromosome, and the definition of a chromatid is this thing right here. So this would be a chromatid, okay? And the question is telling me that I have 16 chromosomes, but I have, and then it's asking me how much chromatids I would have. And so that means I would just have 16 times 2, because for every chromosome I have 2 chromatids. Did that make sense for everyone? So that means I would have 32 chromatids. So that's why the answer is C. Is it SL or HL? This is a SL paper. Someone's asking, does it actually only take 5 days to learn the whole syllabus? Yeah, so we do a crash course with a professional tutor, and she literally goes over everything you need to know. for the syllabus, and you learn everything, and you do practice questions with her, and you get loads and loads of resources. So if you're interested in that, you can go on our website. Okay, perfect. I hope that made sense for everyone. Does it matter that it says about the late prophase? I think it matters a bit because you have to know that in late prophase that there are 16, like there's the diploid number of chromosomes. Okay, perfect. Can you explain this question again? I can go ahead and explain it again. No problem. So this image shows a cell from the root tip of an onion during the late prophase of mitosis. And I know during late prophase of mitosis, I have 16 chromosomes or my diploid number, because that's exactly what it tells me in the question. Okay, and it's asking me how many chromatids are present in the cell. Well, I know that this is a chromosome right here. And by definition, only one of them is a chromatid. So if I have 16 chromosomes, so 16 of those two little chromatids here, that means I would have 16 times 2, which equals to 32 chromatids. Okay, perfect. I think it's good to move on. Okay, question 6. So question 6 says the diagram shows a chemical reaction taking place. How would the reaction be described? So I notice that I have a chemical reaction between sugars taking place, and I know that something is being joined together. I'll give you that as a hint. So something, it's not being broken apart, it's being joined together. So I'll give you 30 seconds to work that out, and then we'll go over it together. D. Which past paper is this? This is the M23 Time Zone 2. D. Okay, good. I'm seeing a lot of D's in the chat, which is really good, because the answer is D. Perfect. If you guys want to go ahead and give reasonings as to why it's D in the chat, maybe to help you practice and try to rephrase think of ways you can phrase the answer on your actual exam. Okay, perfect. So I can see here in my question that I'm joining two sugars together. I'm joining two sugars together, and the reason I know that is because there's no connection here, and suddenly there's a connection here. And what happens when you join things together, that's actually called condensation reactions. And condensation reaction is when you join things together and you use up water. So I already know that my answer is going to involve condensation, because hydrolysis by definition means breaking up stuff. Perfect. So condensation, it would be hydrolysis is wrong and hydrolysis is wrong. And so my answer would be B or D. And then you also need to know that anabolic sure that's on. Anabolic also means joining, so the reaction of joining things together. Okay, and catabolic is the breaking things. Yeah, anabolic is building things up, and catabolic is breaking things down. And clearly here we're building things, we're joining things, so it has to be anabolic, so it can't be D. So for question 6, the answer would be D. It's an anabolic condensation reaction. Perfect. Hopefully that makes sense for everyone, and then we'll move on to question 7. Where is the water released though? Yeah, so in joining, the water is actually used up. So in condensation anabolic reaction, water is used up. Okay, let's move on to question 7. The image shows a molecule of oleic acid. What describes oleic acid? And I'm going to give you guys a hint. You're going to need to focus on this section of your molecule. I'll give you 30 seconds to work that out, and then we'll go over the answer together again. Okay, I'm seeing B in the chat. A, okay. This is an SL paper 1, by the way. B. Okay, this is really good that we're getting a lot of B's in the chat because B is the correct answer. Okay, so the answer is B. And why is that? Okay, let's go into understanding why that is. So, we clearly see here that we have a unsaturated molecule. And the reason that is because we have a double bond. Okay, but let's go over the definitions of cis and trans. Okay, so cis double bonds usually have H's on both sides like this. And so this would be a cis. And the trans actually have H's on opposite sides like this. Okay, let me just make sure. Yeah, trans, this would be a trans. And I can clearly see in my image that my image matches the description of a cis, or the image of a cis fatty acid chain rather than a trans. So I can already cross out the options C and D. Now let's look at further in the question. The difference between A and B is that it says it's polyunsaturated, and then here it says monounsaturated. And does anyone know the definition of poly and mono? So poly is generally something to, yeah, well, mono is one double bond. Poly is many, exactly. Poly is many, multiple. So what you would see in your image is basically multiple double bonds within the chain. But we don't see that in the image. We only see one. And mono basically means one. And because we see one double bond throughout our whole chain, we can say that this is a monounsaturated. So our answer would not be A, it would be B. So good job for everyone who got that in the chat. That was really good. Yes, mono is one double bond, poly is multiple. Perfect. I remember the cis-trans thing with, yeah. Perfect. So someone asked earlier, how would I know if it's cis versus trans? Just look at your molecule and see, are my H's on the same side or are they on separate sides? So do you see that in the image, how they're on like, one is on top and one of them's at the bottom? Okay. Hopefully that explanation made sense. Let's move on to the next question. So question 8. No, this is biology. This is not chem. Wait, can you say that one more time, please? Yes, what would you like me to repeat? The cis and trans, or what would you like me to repeat? Yeah, exactly. You look at the H's. Oh, no worries. The cis and trans are also part of chem. Yeah, it's also part of organic chem. So everyone should be reading question 8 and looking over the options. So question 8 states, what is the proteome of an individual? And I'm going to highlight the word prot, because that gives you a hint at what the answer is. And for this one, I would say, for question 8, I would say you have to memorize the definition of what a proteome is. And the prot part of the word gives you a hint at what the answer is. So I'm getting a C. That's really, really good. A. Okay, perfect. Yes, it's bio-SL. C. Okay, C. 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 Okay, I'm going to give a couple of seconds for anyone who C. Okay, perfect. I'm getting a lot of C's in the chat, which is actually the correct answer. And the proteome of an individual, by definition, is the complete set of proteins expressed by an organism. So, and the way I like to remember that is thinking proteome has the word prot in it, and that's how it helps me remember, oh, it's the complete set of proteins in expressed by an organism. Okay, let's look at the answer options and see how I would use process of elimination to work that out. So A says amino acids unique to an individual making up the proteins in cells. So the key word in this answer would be amino acids. So it's talking about the amino acids, and that's not actually what we're looking for. We're looking for proteins. So it would not be A. Okay, let's look at B. the way in which an individual's polypeptides are folded into three-dimensional structure. Again, I don't see any words really, well, polypeptides relates to proteins, but I don't see a clear um, connection to proteins. So we're not talking about polypeptides, we're talking about proteins. Let's look at C. Proteins synthesized as an expression of an individual's genes, and that's exactly the correct answer. I see proteins, and that's the key word I was looking for in my definition. Okay, so it's not B. Let's look at D just for practice. D states all possible combinations of amino acids an individual contains. So the key word here would be amino acids, and again, we're not looking for amino acids, we're looking for proteins. So that's how I know my answer is not D. Hopefully that made sense. Place any questions in the chat. I believe there's an assistant tutor in the chat that can help you if you have any questions that I don't answer. But yeah, so that's how you would answer 8. Do IB Chem. If a lot of people request IB Chem, we could consider doing an IB Chem one. Okay, perfect. Let's move on to question 9. Okay. Question 9 states, in the grass plant, I'm going to butcher the name, so I'm just going to skip over the name. This is the name right here. Okay, the enzyme amylase, so this is our enzyme, breaks bonds in polysaccharides during germination. The graph shows how the activity of the enzyme varies with the concentration of polysaccharides. So, basically what the graph is showing me is that I'm increasing concentration. How does the enzymes working, how is that affected? And how does that affect the rate of change? The rate of reaction, sorry. That's basically how I dissect the graph, increasing the concentration of polysaccharides, and how does that affect the enzymes? And the question states, what is the reason for the curve leveling off? So it's asking, why does all of a sudden, if we look at our graph, why all of a sudden does it just starts getting straight here? So increasing, and all of a sudden I'm leveling off, I'm becoming straight. So why does our graph do that? I'm going to give you 30 seconds to try to answer this in the chat, and then we'll go over the answer. Okay, I already see a couple of D's. 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 Okay, I see a lot of D's, which is really good because the answer is D. But let's go over why that is, okay? So we have, let's draw it out because I like drawing things out to understand them. We have these enzymes. These are called amylase. So these are my enzymes, and my enzymes, okay? And I have these polysaccharides here and here, okay? And as my reaction is progressing, I'm slowly adding more concentration of polysaccharides. So essentially, I'm just giving in more polysaccharides. So if we look here, we have some polysaccharides joining in the reaction of the enzymes, okay? And then more here. And as the reaction is proceeding, I'm continuing to increase the concentration of polysaccharides. I'm adding more concentration, adding more concentration, adding more concentration. So basically, I'm just drawing this section of the graph, and I'm showing you how if I keep adding more concentration, substrates are going to go ahead and react with the enzymes. Okay, but what happens when I keep adding more concentration of polysaccharides? So I keep adding more, but actually, there's no active site for the enough active sites for my polysaccharides to join on. So even if I keep adding or increasing the concentration of polysaccharides, nothing will happen to my reaction because all these active sites are already being used. So do you guys see how that happens? And this is a process actually called when the I forgot the word, but I forgot the word. If I remember it, I'll say it. But basically, all these active sites are used. So that means even if I keep adding more concentration, my rate of reaction will not change. And that is why our graph levels off. Okay, so let's look over the answers and see why the answer is D. Okay, let's look at A. There's insufficient substrate for the enzyme to act on. Well, that's actually incorrect because I've seen here that I have enough substrates. All these polysaccharides were my substrates. So my answer cannot be A. Okay, product acts as an enzyme inhibitor. There's no mention that any of the polysaccharides are inhibiting my enzyme, so it cannot be B. Okay, the enzymes have all been consumed in the reaction. That's definitely incorrect, and that's one thing you should know and keep in mind that enzymes are never consumed in a reaction. Okay, so it's definitely not this one. Definitely not this one. This one's completely wrong. And then let's look at D. All the enzymes' active sites are occupied by a substrate. And that's why the answer is D. That is correct. And in the drawing, we were able to see that all the active sites were being occupied by the substrate. That's why the rate of reaction couldn't have continued to increase, and that's why it levels off. Did that make sense for everyone? Okay, hopefully that made sense. Let's look at question 10. Perfect. I'm getting a lot of yeses. Okay, question 10 says, what is bonded to phosphates in strands of RNA? So, the phosphate in an RNA strand. I'm going to give you 30 seconds to answer this question, and then I'm going to go ahead and work it out with you. Okay, what is everyone thinking? Some A's, okay. D's. A, D. Okay, so it's between A and D. RNA, so ribose. Oh, that's pretty good. So the answer is actually A. So only carbohydrates. So let's go and explain why that is. So if I go ahead and draw my RNA molecule, so this would be my phosphate. So I'm just going to put a P to represent phosphate here. And then I'm going to go ahead and draw my ribose. And then this is my base. Okay, let's connect that to another. Okay, this would be my RNA molecule. Of course, your RNA molecule will be much longer, but this is just two nucleotides from that RNA molecule. And I see here that this is my phosphate group. Okay, and it's asking me what is bonded to phosphates in a strand of RNA. So if I look at my phosphate and see the connection between phosphate, I see that it's connected to my ribose sugar. And what is ribose? What is a ribose sugar? A ribose sugar is a carbohydrate. And I don't see it connecting to anything else. So the only connection is this one right here. So I know that A must be the right answer because it's only connected to the ribose, and that is also a carbohydrate. So it can't be B, it can't be C, or it can't be D. Did that make sense for everyone? Doesn't matter which side it's on. So you mean if you draw the phosphates on this side instead of the other side? Yeah, it doesn't matter. As long as you... Isn't there a bond linking the phosphates too? Yeah, so you mean this one here? So they're both connected to the right in both bonds. There's two bonds, and then they're both connected to the ribose. Can you repeat it again? Yeah, of course. And no, I'm confused why it's not D. Okay. You can go ahead and re-explain that. Okay, so if I redraw my RNA molecule, let's draw it again. Let's do colors this time. Maybe this will be easier for people to understand. So this is my phosphate group. Let's call that P. Okay, and this phosphate group is attached to a ribose sorry, my drawing is quite bad, but hopefully it's making sense. My ribose, and that is attached to a base. So by base, I mean this is adenine, guanine, thymine, and so on. Okay, and so this would be one nucleotide, and then the way it attaches to another one is like this, and then here we have another phosphate group, and then it connects like that, like that, like that, and then there's another base. Okay, does everyone understand this drawing so far? So these are the locations of my phosphate groups. Okay, and I see, just by looking at it, I see that the only connection or the only attachment that the phosphate group is having is with the ribose sugar. So even if I look at this one, which has two connections, they're both only connected to my ribose sugar. And what do we know ribose sugar is? We know ribose is a carbohydrate, and that is why it's only going to be connected to carbohydrates, and that's why my answer is A. Hopefully that made more sense for the people in the chat who are okay, perfect. Yes, it makes sense. Okay, perfect. So that's why the answer was A. Perfect. Okay, let's move on. Okay, question 11. The anticodons of 3 tRNA and the amino acids they carry are shown in the table. So these are my tRNA here, and these are my amino acids corresponding to the tRNA anticodon. Which base sequence of mRNA molecule would code for these amino acids form a tripeptide? Okay, so I'm going to give you a couple of seconds to answer that one, and we'll go over the answer together. Okay, someone said, what is the difference between the time zone 1 and time zone 2? Are the papers easier for a specific time zone? Well, us IB students always like to say that one time zone is easier. And when I was studying for my exam, I always said, oh, one time zone is easier than the other. But I don't know if IB is actually doing that or not. But there's different time zones is because there's time zones in the world, and people take exams at different times, and they want to prevent people from cheating. So that's why they have two different exams. Yeah, exactly. Just exactly like Basil said. He said, so people can't cheat. Okay. So question 11. I'm getting a lot of D's in the chat. Yes, Kanye West said D. Who else? Laura said D. I think Laura's on a roll, guys. She's been answering most of them correctly. Okay. D, D, D. Perfect. So the answer is actually D. And I'm going to go ahead and explain why that is. Okay. So if we look at let's draw it out, because that's the best way to explain things. This would be, it's a really bad drawing, but this would be my tRNA molecule. So this would be my tRNA molecule, okay? And here I have my anticodon. Okay, and then if I drew out process, this would be my mRNA molecule, okay? And my mRNA molecule would have the codon. So my tRNA molecule has the anticodon, and my mRNA molecule has the codon. Okay, and the question is asking me, what is the base sequence on the mRNA molecule, not on the tRNA molecule, on the mRNA molecule, and what would that code for these specific amino acids here? Okay, so we first look at the first amino acid, this one here, and we look at the tRNA molecule. So on the tRNA molecule, it would have these bases, so it would be G, C, and A. And if I have G, C, and A on the anticodon on the tRNA molecule, that means I'm going to have the bases that are exactly complementary to those on the mRNA molecule. So on the mRNA molecule, I'm actually going to have, so what's complementary to G? That would be C. And then what's complementary to C? That's G. And then what's complementary to A? That would be U. Because remember, there's no thymine in mRNA. So I know to code for my first amino acid, I'm going to have this on my codon on my mRNA molecule. So I can already go ahead and look at my sequences given to me and see which ones start with CGU. So it's definitely not this one, because this one starts with G. No, this one starts with G, with C, sorry, it goes to G, but it actually has T, and that's incorrect because we don't have thymine in the mRNA. We have uracil instead, and we can see option D shows us perfectly CGU. So I know my answer is going to be B. And if I go ahead and went in and filled out my basis for every single one, I would actually see that it matches for this amino acid and this amino acid. So, but for the sake of my B exams, I would just say if this works out well for you, and you don't spend too much time working out the others. If you have more time in the end, maybe come back and see if those work out. Thankfully, it's really easy to see that the answer is D in this case. Which topic is this question based on? So this would be 2.7, I think. So topic 2 for the old syllabus. What question are we on? We are on question 11. Did that make sense for everyone? So let me know in the chat if you need any clarifying questions. Hopefully that made sense for everyone. Okay. Someone sent me a flame heart. I don't know what that means, but thank you so much for sending me a flame heart. How many questions are there? There are 30 questions. Ooh, 30 questions? Yeah, there's 30 questions. So we're almost there, almost halfway. We should be finishing at around 9:30. Will we cover the whole paper? Yes, we will. Okay, let's go on question 12. Question 12 says, which equation represents a process in yeast that causes bread to rise? And I'm going to give you a hint. Okay, that's my hint. Anaerobic respiration. Let's see what answers we get in the chat. I'm getting A's, D's. Fermentation. That's another hint, yes. A, okay. Okay, so I'm getting a lot of A's and D's, and we'll go over the answer. So the answer is actually D, but I can see why you got confused. Some of you got confused with A. That's completely normal. But when we think of anaerobic respiration or the process of fermentation, we think of anaerobic means no oxygen. So respiration that's occurring with no oxygen. That's in comparison to aerobic respiration that does involve oxygen, okay? But in this question, fermentation involves the process of anaerobic respiration, so I know I have to start thinking no oxygen. So I can already look at my answers and see which answers involve oxygen in them. So answer A does have oxygen in it, so I can already cross that out because I know anaerobic is no oxygen. Answer C also has oxygen in its answer, so I can already cross out C. So I've already eliminated A and C, and that's already pretty good. So now it's just 50-50. And now I look at my product side of my equation. For B, it says carbon dioxide and water. And then for D, it says ethanol and carbon dioxide. And actually, the answer is D because the products of anaerobic respiration or the process of fermentation is always going to be alcohol, so ethanol and carbon dioxide. For this here, so the products carbon dioxide and water, that would actually be aerobic respiration. So what I like to think of it is anaerobic respiration is produces an alcohol. That's the process of fermentation. That's the process of making beer. So maybe making those connections in your head. So that's why it wouldn't be B, because those are the products of aerobic respiration. So it wouldn't be B. It has to be ethanol and carbon dioxide. Did that make sense for everyone? So for this process, alcohol is always required. It's not required, but it's a product of the process. So have you ever heard someone say, oh, beer is made from fermentation? So you think like it's a byproduct. No byproduct, it is a product. Did that clarify your question? Okay. Okay, thank you. Okay, perfect. So yeah, so this would be quite an easy question to answer. I would say that it is an easy mark if you know the differences well between aerobic and anaerobic. And so I would just always remember that anaerobic is no oxygen, and then aerobic is oxygen. And someone asks what is the difference between aerobic and anaerobic, and that's basically the presence of oxygen. So I'll highlight that for you here and here. Okay. Okay, now moving on to a difficult question. When I had it was this question also showed up on the HL paper, but it was a bit of a different question. And when that showed up on my exam, I just remember side-eyeing my friend who was sitting across from me because I had no idea what was going on. But I remembered that um, we actually did this topic in the crash course, and the crash course saved me for this question. So, yeah, because I don't know why teachers they love skipping over graphs. I don't know why. My IB teacher never taught us one single graph. Okay, so question 13 states, which graph represents the action spectrum for a green plant receiving only blue light? So I'm going to give you a couple of seconds to answer that in the chat, and then um, we'll go over the answer together. Yes, because so IB never talked about the action spectrum or any of those graphs for the past, I don't know, like 5 years or 10 years of exam papers, and all of a sudden on the M23, they surprised everyone. Okay, I'm getting B's or C's, A's, okay. C, okay. Okay, let's go over the question. So for this question, for those of you who said B, congratulations, that was the answer. And why is the answer B? Let's dissect the question. So it's telling me that it's an action spectrum for a green plant that only receives blue light. Okay, and to be able to answer this question, I would have to understand the graph and what the graph is presenting me. The graph actually is presenting me the rates of photosynthesis graphed over the wavelength. And one thing you do have to know is what the wavelengths represent. So I'm just going to show you in this sample one here. The wavelength actually represents different colors. So from this wavelength to around here, so I would say like this is blue light. Okay, and then I think that's a bit too much actually. But around here, that's blue light. And then from this wavelength to this wavelength, that's green light. And then finally, from this wavelength to this wavelength, that is red light. And the way I like to remember it is that red lights have the highest wavelength and blue have the shortest wavelength. Okay, so that is something you would have to know or memorize, or maybe if you take chemistry, this could be easier. How can you tell? I think you just have to be exposed to the question. You just have to know that red, red light or red has a higher wavelength, the highest wavelength, and then blue. If you think about a rainbow and like red, orange, yellow, the red has the highest wavelength, and then the other end of the rainbow would be the lowest wavelength. So if you would want to remember it like that, and then remember that okay, red would be on this side and blue on the other side. So that's how I would how I could tell when practicing for my exams. So now that I identified which wavelength corresponds to which color, and I see that the question is asking me for a green plant only receiving blue light. And let me look at my answers. Let me look at my answers. When you have a peak in the graph, that means that that's the color that the plant is receiving. So I can see for option A, the plant is receiving blue and red. Okay, and then let me go look at option C. Option C is clearly receiving green. Now let me look at option D. Option D is again receiving red and blue. Okay, and option B is actually the only answer that shows me there's only receiving blue light. So that's why my answer would be B. That makes sense for everyone. Let me just scroll up the chat to see if I missed any question. Is this chem or bio? This is bio. It's an SL paper. Okay, so you would say those 10 extra questions, they would be C, C. Yeah, so those of you who said B, got it right. You don't understand why C is a green. Basically, you just have to memorize that this position here is a blue wavelength, this position here is green, and this position is red. And it's not something you would know if you haven't seen the question before, so I understand why, but I'm telling you now, know that this is blue, this is green, and this is red. And then why is C green? Because this is the green wavelength. So this positioning between here and here is green. Is it always blue, green, red? Yes. Yes, it's exactly. So just me, 2004 answered it perfectly. Yes, exactly. Exactly. So, I wouldn't worry too much about the number. I think Chris is asking, does it start at a certain number? I wouldn't worry too much about the number. Just maybe remember it visually, or remember that it's always going to be blue, green, red. So we need to memorize what the different wave colors look like on the graph. Not what they look like on the graph, but their positioning. So you just remember that this is my, let's call this blue and green and red. And again, I'm not looking at a specific number. I'm just looking at like, let me divide it in 3 and split it up 3 equally. Is this bio? I just joined. Yes, it's bio. This is HR. This is SL. Okay, perfect. I'm going to move on to the next question. Hopefully that made sense for everyone. Okay, what change causes sickle cell anemia? So we're on question 14. It's asking us what change causes sickle cell anemia. And this change is something you actually have to know or have to memorize. Yeah. Give you a hint. It has to do with the mutation. So I'm getting C's and D's. C, D, B, B. Okay, so I would say I was getting mixed up answers. B or D, C. Okay. For all of you who said C, you are correct. That is actually the correct answer. And let's go ahead and explain why that is. C, but don't know why. Yeah, same. Okay, so let's look at A. So actually, let me go explain what causes the sickle cell anemia, and then we'll go over the answers. So what happens that causes sickle cell anemia, and this is actually something you have to know by heart, is that there is a change in the amino acid sequence in a polypeptide chain. So the amino acid. Oh, hello, can you guys see me? For some reason, I just got a weird notification. But okay, so hopefully you guys can still hear me. Hello? Yeah, perfect. Okay, so the amino acid changes from glue to val. So that's something you do have to know, is that it changes from glue to val. So glutamic to perfect. And I would say memorize that for your exam, the change glue to val. And then also memorize that this change is caused by a change in the mRNA molecule of the codon going from GAG to GUG. So these are the two things I would memorize to know that these are what cause sickle cell anemia. Okay? And if we look at the answer options, now let's dissect the answer options. Okay? Let's look at the answer options and see what connects to this amino acid changing because of mutation and also leading to the caused by the mRNA molecule going mRNA codon going from GAG to GUG. Okay, so let's look at option A. Option says one amino acid less in a polypeptide chain of hemoglobin. Okay, um, I see where you might get confused with this option A because, again, we talked about amino acids, but it's not about it being one amino acid less. It's still an amino acid is there. It just changes from glue to val. So the option is not A. Okay. B, a mutation leading to an extra codon in the genome. Okay, I see where some of you might get confused with that because it does say mutation. But again, it's not leading to anything less or extra. It's just causing a change. So it's not going to be B. Now let's look at C, thymine replacing adenine in DNA. Okay, and C is actually the correct answer. And why is C the correct answer is because we have previously memorized that the change goes from GAG to GUG, right? And if the change on the mRNA molecule says GAG to GUG, I can then conclude that my DNA molecule is going to have complementary bases to that on the mRNA molecule. So on my DNA molecule, it's actually going to go CTC to CAC. Okay, so this information I've pre-memorized, but this question is making me reflect and use my applied knowledge from topic 2 and connect the fact that mRNA and DNA have complementary bases, and I can see that my thymine actually replaces adenine in DNA. So that's why my answer is C. Okay? Hopefully that made sense for everyone. And then 14 says failure of tRNA to correctly transcribe the sequence of codon from mRNA, and that's incorrect. There's no issue with tRNA. Hopefully that makes sense for everyone. Let me know if it doesn't, and then we'll go ahead and move on to question 15. So the image shows a human karyogram. For which person was the karyogram obtained? I'll give you 10 seconds to answer this in the chat. And then we'll go ahead and work out the answer together. Can you zoom out a bit? Yeah, of course. Is that better? Okay, I'm getting B's and D's in the chat. B, 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 B. B. Okay, perfect. So the answer is actually B. And let's go ahead and explain why that is. Okay. So the question options are asking me to identify whether this karyogram is male or female. So let's go ahead and do that first. And to identify that, you have to look at these chromosomes here. And one thing you do need to know is that when you have two XX chromosomes, so when I have an X and an X, that means I'm a female, right? And when I have a X and a Y chromosome, so let's say an X here and a Y, and the way you'll see whether it's a Y chromosome, you'll see that the Y chromosome is actually much smaller. So this would be an X and a Y chromosome. That means I'm biologically a male. Okay, so I have female presented like this to me, and then male presented like this to me. And now I can look at my karyogram and see that I have two chromosomes of equal size and equal length. So I can make the conclusion that these are two X chromosomes. So if they're two X chromosomes, I can say that this is a female. Okay, and I can go ahead and cross out C and D because they say that this is a male. Now let's look at the second thing. It says with or without Down syndrome. And how do I identify if the person in the karyogram has Down syndrome? Well, I have to look for something called non-disjunction. And that occurs in meiosis when the chromosomes split up and they don't split up properly. And that causes Down syndrome or trisomy. And you'll see the word tri in trisomy because you'll see that if I had Down syndrome on chromosome 21, on chromosome 21, you would see instead of 2 chromosomes like you would see presented here, you would actually see 3 chromosomes. Okay, so I look at my karyogram and see if there's 3 chromosomes placed together rather than 2. And then I see 2, this one's 2, this one's 2, 2, 2, 2, 2, 2, 2, 2. Perfect, 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 perfect. There's no non-disjunction happening. There's no 3 chromosomes paired up together. So I know that this patient or this person does not have Down syndrome. So the answer is a female without Down syndrome. Did that make sense for everyone? Chris says, I'm back with food. I ran. And now my dog is hyped. Yeah, perfect. Okay. Okay, let's move on to question 16, which is an inheritance question. And these questions I personally hated in my exam. These were the worst. If this showed up on my exam, I would silently shed a tear. And I don't think this was on the HL paper. So I was lucky for that, but this was on the SL paper. I'm going to read the question, give you 30 seconds, and then we'll work it out together again. Okay? The pedigree chart shows the inheritance of 3 generations of Duane syndrome, a condition caused by a dominant allele that affects the alignment of the eyes. Okay? In individuals 2-1 and 2-2, if they had a third child, so question mark, what is the probability that that child would have this syndrome? Regina says she hates them too. Yeah, I hate them too. Yeah. Okay, so the question says, if they, hypothetically, this person here and this person here had a third child, what would their, the possibility of that child having the syndrome be? So I'm going to give you a hint. I would draw a chart to try to dissect this question. I'll give you 30 seconds and we'll work it out together. Okay, I'm getting a B. Chris is doing a B. Set a B in the chat. I never get the stuff right, yeah. A, okay. C, 50%, which is B. So the answer is actually B. Hopefully I gave you guys enough time to work that out. Okay, and I'm going to show you the exact steps I go through when solving inheritance questions and how that helped me get all of them correctly. Okay, so when I look at my diagram, at my pedigree chart, I first identify everything I know. So what do I know? I know that all the squares and circles in white are people who don't have them. There are people that don't have them, and the question is telling me that this is a dominant allele. So if it's a dominant allele, that means everyone who doesn't have the disease, it has a recessive allele. So they have, let's say, R and R. So we'll label it as R. So I think that's an easy way to identify the difference between recessive and dominant. So this person would have R and R are recessive. This person would have R and R recessive, and this person would have R and R recessive. Okay, I've identified which ones don't have the disease, which is good. Now, let's try to fill in the ones that do have the disease and try to fill in the alleles that they do have. Okay, let's look at this person here. So if we drew a chart, we know that the female is recessive, and let's try to figure out what that is, what the man is. So the dad has a dominant allele. We don't know. He could be R, or he could be R and R. So these are the 2 options that the dad can be. So I'm going to try to draw 2 different charts and see which one matches the set of children he's going to have. So let's do option 1, where the dad is capital R and capital R, so they're both dominant, so R and R, and let's go ahead and draw that out. So R, 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 R. R. And what I actually see is if the dad hypothetically was two capital R's, that means all my kids have 100% chance of having that disease. But I can see that actually one of the kids didn't have that disease, and one of the kids had a recessive allele. And so that means dad could have never been a capital R, have he could have never had that as a dominant allele, because that means then his kids would just all have that disease, but that doesn't make sense. So I can then reflect and say, let's look at the other option. And the other option that the dad could be is capital R, small r. So I know that it can't be two capital R's, so it has to be the other option. So let's draw that out. So R, capital R, small r, small r, small r, small r. Okay, and I can see from this chart much better that there's a 50% chance of the children having the disease and a 50% chance of the children not having. And that makes more sense because I see a child that didn't end up having that disease. Okay? So hopefully, why is it not RR instead of RR? So again, so hypothetically, so let's draw in a different color. Hypothetically, if the dad was capital R, like this, so if the dad was like that, that means that all the children would have had that disease. Does that make sense? Because it's a dominant allele, so all these kids would have that disease. But I can see from the chart, the pedigree chart, that one of the children actually doesn't have that disease. So it's impossible for it to be this option. Hopefully that made more sense. Why it's not capital R. Okay, so it has to be this here. Capital R, small r. So we know that the dad, now we've identified that the dad is capital R, small r. And then we know that his kid who has that disease is also going to be capital R, small r, because those are the only options. So I know that this kid here is capital R, small r. So now I've identified individual 2, which is what we're looking at here, is capital R, small r, and individual 1 here is small r, small r. And now I can just draw a chart again to see what's the percentage of that child having that disease. So R. And I can see from my pedigree chart that 2 out of the 4 boxes are filled with the child having that disease. So that means 2 out of 4, or 1/2, or 50% chance of that child having the syndrome. Does that make sense? So that's why it's going to be B. Okay. Hopefully that made sense for everyone. Let me know if you have any more questions and I'll go over it again. But if that made sense for everyone, we can move on. Mia got it. I'm proud. Can you please repeat the explanation? Of course. Is everyone okay with me repeating the explanation, or do you want me to just move on? Because the recording will be online, so in case you need to rewatch it. So just tell me yes or no if you want me to repeat it again, or no, move on. And then hopefully the person that requested it, please repeat it. Yes, it's okay. I can repeat it. Okay, let me repeat it. Yes, okay. I'm just gonna start from square one, if that's okay with everyone, to repeat it for everyone. So when I have these type of questions, I always identify what I know. Okay? And the first thing I know is that this disease is a dominant allele disease. That means that people who have that disease are individuals who have an allele RR or R small r. Okay? And I know people who don't have the disease or unaffected have to be recessive small r. Okay? Because this is a dominant allele. So these are the things I know. Now, let me label that into my chart. So the next step would be labeling in the chart. I know that this is small r. This one's also small r. And this one's also small r, because I know all these individuals are unaffected. Now, let's go to the harder step. I think this would be the hardest step in the whole question, is trying to find out what these individuals are. Okay, I think my screen is lagging. Yeah. Oops. I'll give it a second. Apologies, guys. My screen is lagging. Well, this is our first time doing this, so there's a bit of tech issues. Um, okay, let's say this is your chance to get a 5-minute break while I try to work out these tech issues. And then we'll come back to answering the questions as soon as we can. So go have a stretch, get a 5-minute break while I work out the tech. And hopefully this doesn't keep happening. Okay, is the screen showing up well for everyone? How often do we do these live streams? So this is the first time we've done this. Um, so which explains the tech issues. Okay, there you go. Apologies again for this. If this does become a regular event, I think we'll try to work out more how we can prevent this from happening in the future. So let us know if you guys like this and if you want more of this. Okay, perfect. We're back to square one. I'm happy that this is very helpful. Thank you so much, Mia. Okay, everyone sees the screen. It's perfect. Can I move on? Chem paper 2. Okay, we also do chem papers, so we can. We want more. Okay. So I'm going to go ahead and continue. Okay. So we stopped at identifying that these individuals are all recessive because they don't have the disease, okay? And then the hardest part would be identifying what these individuals are here, right? Because it's much more difficult because they could be either option 1 or option 2, which is capital R, or capital R, small r, okay? So the way I would identify which one is which is just by trial and error. I would try both and see which one matches more the pedigree chart. So let's try option 1. So let's call this option 1. And let's say hypothetically, this dad right here was big R, big R. So let's draw the chart out. Okay. And the mom, of course, is RR, which we already decided a long time ago. Okay. So we can go ahead and fill in the chart. So R, R, large R, small r. And if we go ahead and film, oh, the stream on YouTube is not fixed. Okay. Let me go ahead and fix that right now. Thanks for the okay. Just let me know when it is fixed so I don't. Okay, is that good for YouTube? Okay, I think the stream is okay. Okay, perfect. Okay, let's just continue. So, again, sorry. So, individual 1, the dad, we said he could be option 1. So, it could be capital R, capital R. Okay, we tested that out and we found out when we drew the charts that if the dad hypothetically was capital R, that means that all the children they would have had to have had the disease because they have all the dominant R, the capital R. Okay? So we know that that cannot be a possibility. The dad cannot be 2 capital R's because from the pedigree chart, we see that one child actually does not have the disease. So one child does not have the disease means that the dad can't be 2 capital R's because if he was 2 capital R's, that means all his children would have had the disease. So I can eliminate option 1 from being an option, and we can go ahead and try option 2. So if the dad hypothetically was capital R small r, like this, and of course the mom is recessive, and then I go ahead and draw chart, that makes much more sense because I can see that there's a possibility of the child either having the disease, so this section right here, or not having the disease, this section here. Okay, so that's how I know that the dad cannot be capital R, he has to be this here. So I went ahead and identified that the dad is capital R small r. Okay, and I know that if he had a child with that disease, then that child had to have been capital R small r, as my chart says. So that means individual 2 is going to be capital R small r. Okay, and we know that individual 1 here is recessive, of course, because they don't have the disease. So let's go ahead and finally draw the chart that represents individual 1 and individual 2, and find out what is the probability if they had a third child. What is the probability of that child having the disease? So R and R for the mom, and R for the dad. And just filling in my chart, I can see that the probability of the child having the syndrome is 50%, because two out of my 4 squares show that they can have 50% chance of having that dominant allele, and these ones here would have no syndrome. Okay, turns out to be easy. Thanks. Okay, perfect. I'm glad that made much more sense. Okay, perfect. So that's why my answer would be B. So identify what you know, what you don't know, and then trial and error, find out what the answer could be, and then fill in the blanks. Okay, perfect. Now we can move on to question 17. Question 17 states, what is combined in the production of cloned embryos by somatic cell nuclear transfer? Is this SL? Yes, it is. Okay, I'm going to give you a couple of seconds to work this out. So, production of cloned embryos by somatic cell nuclear transfer. So, a somatic cell is just a cell that is tissue cell or a organ cell. Yes, the recording of the live will be on YouTube, and we're actually also live on YouTube now. So, yeah. Cloned embryos is basically when you have you heard of like cloning the sheep dolly? Is when you create an exact copy of an organism or an individual. So, for example, they cloned a sheep dolly, which means that they made 2 times of the same exact person, so the same DNA, the same everything. Have you seen those movies where they talk about like clones? Let's see if I can find a proper definition. Cloned, the scientific definition. Okay. An identical copy. That's basically, so the scientific version didn't help that much, but it's identical copy. So making an exact copy of that. Sorry, is this grade 11? Yeah, if you take IB, yes, it is grade 11. Okay, I'm seeing B's, I'm seeing A's, we don't know. Okay, so let's go over the question. So what is combined in the production of cloned embryos? And this is just a process that you have to know. And actually, I really liked our tutor's explanation in the crash course. So of how she explained this topic. But basically, to clone an embryo, what you do is you take a somatic cell or a tissue and organ cell, and so this is my tissue and organ cell, and I take my nucleus out of it. Okay. And then sorry, I mixed it up. So this is actually my egg cell. So I take my egg cell, so an egg cell from a female, and I remove the nucleus of my egg cell, so that can go away. And what I do is I take a somatic cell nucleus, so now this is my somatic cell, and I take that nucleus here, and I go ahead, and implant that in my egg cell. So a somatic cell is a tissue or an organ cell. Okay, so I take my egg cell, remove the nucleus of the egg cell, take the nucleus from my somatic cell, and put it in the egg cell, and then it's fertilized, and then it grows a baby, of course, and that creates a clone copy. So if I look at my options, based on that explanation, I know that my answer is going to be A, because I have an egg cell without a nucleus, and the nucleus of a somatic cell. So it's not any of these, not any of these. It's going to be A. Did that make sense for everyone? It did? Okay, perfect. Um, yes, okay, perfect. Let's move on to the next question. This next question says, the diagram shows the digestive system of an earthworm. This is the scientific name. I'm not going to say it because it's going to sound weird. Okay, it feeds by ingesting dead organic matter. So it feeds by ingesting, so taking in dead organic matter. What is the mode of nutrition of the earthworm? Give you 20 seconds to answer this one because I think this one's quite easy. Okay, I see an A in the chat. A. The answer for the last one was A as well. And then A. Perfect. I'm seeing a lot of A's. That's really good. Okay. So, let's go over the answers. I'm seeing answers options with heterotrophic and autotrophic. So let's first identify if my organism is heterotrophic or autotrophic. An autotrophic organism is something that makes its own food. So when I think of autotrophic, I think of plants because they basically photosynthesize to produce glucose. They make their own food. So that is what an autotrophic organism is. So my earthworm here is not autotrophic because it ingests other living things for food. It doesn't make its own food, it eats up others. Okay, so we know that it's heterotrophic. And then let's look at the other options. So it's gonna my option A says detrivores, my option C says consumer, and then option D says saprophytes. I don't know how to say that very well, but that's what my option D says. And I know that my answer has to be A because it ingests dead organic matter. And by definition, that's what a detrivore does. It ingests dead organic matter. And a consumer feeds on living. So it's definitely not C. And then option D, these are actually organisms that they don't ingest stuff. They actually secrete enzymes and digest outside the body. So they like spit out enzymes, they digest, it breaks up all the food, and then they like absorb it back in. So that's how I know it's not B. Does that make sense? Hopefully that makes sense for everyone. We're gonna move on to question 19. Which statement applies to transfers in an ecosystem? So A says green plants can transfer heat energy from the sun into chemical energy. B says the greatest loss of energy occurs towards the end of the food chain. And then C says production of carbon dioxide by respiration results in loss of biomass in the food chain. And then D says both energy and nutrients are finite and must be recycled. I'll give you a couple of seconds to put an answer in the chat, and then I'm getting A's. A couple more seconds, and then we'll go ahead and answer. Okay, A. Okay. Unfortunately, the answer is not actually A, but I can see where you got confused with that. The answer is actually C. Okay, transfers in the ecosystem. And the answer is actually C. Exactly, just like Shanae said, plants utilize light energy, not heat energy. Exactly. So, answer A says green plants can transfer heat energy from sun into chemical energy. That's incorrect. Make sure you read the wording of the question. Green plants actually transfer light energy into chemical energy. So that's why A is not correct. Let's look at B. The greatest loss of energy occurs towards the end of the food chain, and that's actually an incorrect statement because um, the transfer of energy between each section of the food chain is actually equal. So you don't lose more energy towards the end or towards the top. It's actually equal. So that's why B is not correct. That's just an incorrect statement. Okay, production of carbon dioxide by respiration results in loss of biomass in the food chain, and that's actually the correct answer because maybe you've seen the little cycle in class, and oh, actually the cycle's here. But we can see that when cells or animals respire, CO2 is released, and that causes biomass to be lost within the food chain. That's why the answer is C. Let's go ahead and look at answer D to eliminate why that's incorrect. And D says both energy and nutrients are finite and must be recycled. And that's actually incorrect because energy cannot be recycled. It is correct that nutrients can be recycled and they can be recycled and are finite. But energy cannot be recycled. Energy is lost. And that's actually what explains option C, because option C says that energy is lost through respiration, and that is correct. So hopefully that made sense for everyone. We're going to go ahead and move on to question 20. Question 20 states that the diagram shows the carbon cycle. Which letters represent respiration? Okay, I'm going to give you a couple of seconds to put an answer in the chat. An Oxford bio student said, "This is so good for procrastinating my uni work." Okay, I'm going to give you a couple of seconds to put in answers in the chat, and then I'm going to go ahead and answer. Thank you very much, Miriam. Okay. So, which letters represent respiration? So respiration, again, remember, respiration is the process of creating energy. Remember, in the mitochondria. I think that's topic. Oh, I don't remember what topic it is. Topic 2, maybe? Okay, so the correct answer for this question is actually D. And we know that plants respire, animals respire, and they respire to make energy. They make ATP. Let's look at the letters and identify which ones are which to help us know why it's D. Okay, let's look at P. Let's look at P. No, I'm sorry, let's look at Q. So green plants containing carbon compounds going into fossil fuels containing carbon compounds. Q is actually incorrect, and the reason that is because plants or organisms turning into fossil fuels means that they just have to die, and then they become fossils, right? So that's not the answer. That would be death, or do they turn into fossil fuels? So Q is not the answer. So any option with Q is incorrect. Let's look at T. So green plants going into animals. Well, animals eat green plants, so that's what represents T. So plants being consumed by animals. That's not respiration, that's just eating. So answer cannot be A. Now let's look at P. P says fossil fuels containing carbon compounds, and that goes into atmospheric CO2. So if you didn't know, fossil fuels actually release CO2 slowly, and that's what P is representing. But that's not respiration, because respiration occurs in living things, and fossil fuels are not living. They're like bones or organic matter that is left over. So it cannot be P. So definitely not P, so we can already cross out B. And then that leaves us with D or R and V. And let's look at R and V more specifically. So R shows that green plants releasing CO2 in the atmosphere, and that makes sense because green plants actually respire for energy. So R makes sense. That is the process of respiration. And then let's look at V. Dead matter containing carbon compounds into atmospheric CO2. And I saw someone mention it in the chat. That's just basically decomposers respiring and releasing CO2 in the chat. Hopefully, in the atmosphere. Hopefully that makes sense. And that's why the answer would be D. let's do question 21. And this is a chi-squared test. And my teacher, personally, my teacher in class never also taught me this. Someone said, still have time to practice every day. A past paper a day keeps the IB at bay. Someone should trademark that. That's pretty good. Okay, so question 21 states, a student wanted to know whether the density of buttercup flowers in 2 fields was the same. He used a quadrant to estimate the number of flowers in equal size areas of each field. The table shows the result. So in field 1, this is how much he expected of flowers to be, and this is how much flowers were actually present. And then in field 2, this is how much flowers he expected to be, and then how much flowers were actually present. Okay? A chi-square test was carried out to determine whether the density of buttercups was the same in both areas. The chi-square calculated value was 4.571. He drew out the probabilities, okay, and then the degrees of freedoms here, okay. With reference to the probability table, what conclusion can be drawn about the null hypothesis with 95% confidence? I'm going to give you a couple of seconds to answer this in the chat, and then I'm going to go ahead and explain that. Okay. If you don't know, I guess. Oops. About to close. If a chi-squared has no haters, then I'm dead. Okay, let's just go over the question. Basically, to find out what conclusions can be drawn with 95% confidence, I have to look at my 0.05% level. Okay, and the reason that is because 0.05 is the same thing as saying 5%. And to look at a 95% confidence level, I would just have to look at the remaining 5%. So that would be the 0.05. So to analyze this graph, when this comes up, and it will always be 0.05, by the way. So just think when chi-squared, I have to look at the section that says 0.05, so I know it's that there. Okay? And then, how do I know which degrees of freedom to look at? Is it degrees of freedom 1 or 2? Well, with chi-squared tests, you always look at the degrees of freedom minus the number of freedoms you have. So here, we have 2 fields. So that means when analyzing my data, I'm going to look at the degree of freedom 1. Hypothetically, I have 20 fields. That means I'm going to look at the degree of freedom of 19. Okay? And same thing, if it was 18, I would look at 17. So now I know that I have to look at 0.05 and the degree of freedom of 1. So I go at 0.05, and then I go to the degree of freedom of 1, and then that gives me 3.841. So I know that it is rejected as 4.571 is greater than 3.841. So my answer would be B. Okay, why degree of freedom 1? You look at the degree of freedom that is 1 less than the number of freedoms you have. So in this case, I have 2 because I have 2 fields, okay? And if I have 2 degrees of freedom, I always look at 1. If I had 20 degrees of freedom, hypothetically, I always look at 1 less, so that would be 19. Yep, that makes sense. Okay, perfect. So that's why I know it's going to be B. If we look at the other answers, we can actually see that A is trying to trick you into choosing this option here, which is incorrect because we know that the degree of freedom has to be 1. So they tried to trick us, but we didn't fall for it. Let's look at C. C and D tried to trick us by making us look at the 0.5 probability level, but we know 0.5 is the same thing as saying 50%, and that doesn't make any sense when looking at a 95% confidence level, so it has to be 0.05. So these two try tricking you into confusing you into looking at the 0.5, and that's incorrect. Let's move on to the next question. We're almost done, just 8 more questions to go. No, it's been long. Yes. The null hypothesis is always rejected, exactly. Just like E. Garcia said. Move back, please. Oh, sorry. Like that? I want to take a pic. Okay, take a pic. Let me know when I can move forward. Is that good? Thank you. Okay, perfect. Okay, question 22. We only have 8 questions left. We're almost there. I know it's been long. Um, okay. Which example of evolution by what? Sorry, which is an example of evolution by selective breeding? Okay, so selective breeding. I'm going to give you a couple of seconds. Maybe I'll read the answers out loud. So A says selection of prey animals that can run faster than their predators. Okay. The variation in the size of different breeds of dogs. Okay. The tendency during breeding for birds to produce more offspring than will survive. Some female spiders only breeding with males which make the right signals. Okay, I'm seeing a lot of B's in the chat. And that is correct. The answer is B. Let's look at quickly at the other answers. So selective breeding is a part of is something is called part of artificial selection. Okay, selection, and that is because it's how humans purposely pick out organisms with favorable characteristics and then make them together. So you can see that in dogs or horses. So if you want a horse that runs faster, you can take 2 horses that generally have fast, and then you breed them together, and that leads you having faster horses as kids. So that's what selective breeding. It's a part of artificial selection, so it's actually man-made. It's by humans. Option A says selection of prey animals that can run faster than their predators, and that actually is not artificial. No humans are involved in that. That's natural selection. Right, because no human is selecting which animal is a prey or which is a predator, so option A doesn't make sense at all. Okay, so it's definitely not A. The variation size of different breeds of dogs, that makes sense because humans actually pick out the different sizes of dogs and breed them together depending on how they want them. So B makes sense. Okay, tendency during breeding for birds to produce more offspring than will survive. Again, that is nothing artificial. No humans are being involved in that. That's natural things happening. So it's definitely not C. Okay, and then some female spiders only breeding with males, which make the right signals. Again, that's not artificial selection. So it can't be D. Okay, let's look at question 23. Almost done. Which statement best describes how evolution occurs? Option A says species which produce the most offspring are favored by natural selection. Okay. Mutations in somatic cells are passed on to offspring. Natural selection decreases the frequency of unfavorable characteristics, and changes in species leads to towards greater complexity over time. So I'm already seeing a lot of C's in the chat. C, C, C. C, yes. Vivian, yes, exactly. Angel, yes. Chris, yes. Leona, yes. Okay, perfect. The answer is actually C. And let's go and process of elimination to understand why it's C. Okay, species which produce the most offspring are favored by natural selection. If you really think about that question, if you really deep that question in your mind, it basically says that if a organism has more children, it's going to be more likely to survive by natural selection. Well, that doesn't make any sense. Just because you have more children doesn't mean you're more likely to survive. Again, natural selection is about those favorable characteristics, who has the characteristics that are favored enough to help me survive against weather issues, against starvation, against, let's say, if an organism is fast. So this doesn't make any sense with connection to natural selection because it's all about favorable characteristics and not about who has more kids, right? And then B says mutations in somatic cells are passed on to offspring. This is actually an incorrect question. An incorrect statement because the mutations that are passed on to offspring have to happen in the sperm and in the egg cell. Okay, so in somatic cells, mutations in somatic cells are not passed on to offspring. So that's someone already moved on to the next question. Yeah, exactly. So B is not correct. And C, natural selection decreases the frequency of unfavorable characteristics. That is correct because again, natural selection is all about how favorable characteristics are more likely to survive, and then how those changes evolve over time. And then if we have the favorable characteristics increasing and increasing, that means the unfavorable characteristics are going to decrease. So that's why C is correct. Okay, changes in species leads to greater complexity over time. You can see why you might get confused with that, because maybe you're making a connection with a specific animal that you have seen evolved, but that answer is actually incorrect. And whenever you see greater complexity, I would say just always think that's a big no-no when you're looking at IB questions, because if you think about it this way, let's say a species evolved to turn green so it can hide better, okay? And just because it turned green, that doesn't necessarily mean it's getting more complex. It's just um, the species that were green were more likely to survive, so these favorable characteristics were passed on. So they didn't get more complex, but that characteristic just were passed on to their children. Does that make sense? Because they survived. So it cannot be D. Question 24 says, the image shows a Tasmanian giant crab found off the coast of Australia. Which phylum does it belong to? So this is one of the things where you have to memorize, and I'm already seeing a lot of correct answers in the chat. Yes, Leona. Yes, user 3567884. Yeah. Any tips on memorizing a different phylum? The way I did it was flashcards, and that actually helped me a lot. And then I would do flashcards, and then I would draw it out from memory. So I would take a blank piece of paper and then draw it out from memory, and I did that like twice, and it was really easy. So always draw stuff from memory, so active recall, that's really helpful in memorizing. Okay, B, Angel got it correct. Perfect. Okay, so what phylum does it belong to? That is an arthropoda, and the way I can see that is because it has an exoskeleton. Okay, it actually has a bilateral symmetry, and it has jointed appendages. And those are all definitions of or characteristics that belong to that phylum. Okay, and if we look more at the others, we can see that this one actually has radial symmetry. So that's, for example, like a jellyfish. So it's not this one. This one's is snails, and those have visceral masses. So it can't be this one. And then this one is like worms. So I like to remember them with animals to help me remember. Okay, and they are long, have segmented bodies, etc. So that's why. What's the difference between the different symmetries? So bilateral means that if you like hypothetically took the animal and like folded itself in its half, it's gonna have the same features on both sides. So one hand here, one hand here. Oh yeah, I can move out the crossing out, of course. Like that. Which topic is this a part of? Yeah, this is topic 4, ecology, I think. Because topic 5 is genetics. Okay, I'm gonna scroll. Hopefully that's okay. Okay, question 25. We're almost there. Can I zoom in a bit? Yeah, of course. Here you go. I'm gonna give you like 2 seconds to screenshot and then I'll move on. No, evolution is topic 5. Oh yeah, my bad. My bad, guys. Yeah, that works. Okay. So the diagram shows a cladogram for a part of the animal kingdom. And I hated these type of questions. These type of questions generally, but okay. Let's move on. Which group does not represent a clade? So a clade, by definition, is a represents this. A clade is all the organisms connected together, plus, that's the key one, their common ancestor. Okay, I'm going to give you a couple of seconds to answer this one. And then I'll go ahead and explain it. So just put in some answers in the chat. An example of radial symmetry is a snail or a jellyfish. I can't really show them right now because I'm sharing my screen on my iPad, but maybe on another device, like search up snail. Okay, I'm getting D's in the chat. B. Okay, we're almost there, guys. We have just 5 questions left. Pretty good. Most of you have been answering them correctly. D, C, D. Okay. Mia is actually correct and May C is actually correct because the answer is C. But let's go ahead and explain that. I also found these questions quite difficult when I was studying. Okay, so a clade is an organism and its common ancestor. And the keyword here is common ancestor. So it's saying that which one does not represent a clade. And it says, option A says 1 to 3. So 1 to 3, so all these organisms right here, okay, all these organisms right here actually do represent a clade because I've highlighted all the organisms and I can see that they all have a joint common ancestor. And it's this point here. So option A is actually a clade. So it's not option A. Okay, let's look at the other options. Other option says 1 to 5. So 1 to 5, this one, this one, this one, this one, all up to 5. And I can see they actually do have a common ancestor, which is this point right here. That's what connects them together. So that is also a clade. So it can't be B. Let's look at option C, which is our correct answer. Option C says 4 to 7. So 4 to 7, they actually have no common ancestor. If they did, there would have been a connection connecting them like this. Okay? You could... I thought it was a clade. Yeah, exactly. So this is the common ancestor that connects... oh no, sorry. Hypothetically, if this was a clade, there would have been a common ancestor here, but because we don't see any common ancestor, it cannot be a clade from 4 to 7. Hopefully that made more sense for everyone. So that's why the answer is not C. And then let's look at 1 to 10. So if I highlighted all of them 1 to 10 right here, you can see they're all connected with a common ancestor here. Okay, hopefully that makes sense for everyone on why the answer is C. Just let me know if you need me to repeat anything. But if not, then we'll move on. Yep, perfect. Okay. Question 26, almost there. Question 26 says the diagram shows the human digestive system. Where does lipid digestion begin? Already got a B in the chat. B. Someone said, isn't it in the small intestine though? Okay, perfect. The answer is B. You are right, it does occur in the small intestine. And if you look at B, it's actually the beginning or the connection between the stomach and the small intestine. So you are correct to say that lipid digestion does occur in the small intestine. Let's look at the other options. So A, it's clearly not A because A is the liver. And no digestion occurs in the liver, but what does occur in the liver is bile production. So you could have got that confused because bile does break down lipids, but it doesn't occur in the liver. Now D actually represents the large intestine, and large intestine, no digestion occurs in large intestine, it just absorbs water. Okay, and then C is not correct because C is the appendix, and again, no digestion occurs in the appendix. Okay, does that make sense for everyone? Is this HL or SL? This is SL. So I'm going to move on to question 27. And question 27 is a bit of a difficult one. But question 27 states, the diagram shows the pressure changes in the left atrium, left ventricle, and aorta during part of the cardiac cycle. Are the valves between the atria and the ventricles open or closed at X, time X, and time Y? Someone said this is their favorite topic. Okay, So I'm gonna give you a couple of seconds, try to give answers in the chat, and then we'll go over it together. Leona says, oh my god, y'all, I'm failing. You're not gonna fail. Don't manifest that into existence. You're gonna do perfectly fine. I'm gonna explain it until you understand it, Leona. Okay. Olive says C, B, and C. C. Okay, I'm gonna go ahead and explain, and the answer is actually B in this case. And I'm gonna go ahead and explain why. Okay, so I'm just gonna draw out my heart. It's gonna be a really bad drawing, but this is gonna be my heart. Okay, and it's asking me, are the valves between the atria and the ventricles open? So I'm gonna look at this valve right here. So this is the valve I'm specifically looking at to see if it's open or closed. Okay, of course there are other valves, but I'm currently right now focusing on this valve. Okay, before I go and explain if they're open or closed, one thing we have to know is that when I'm saying something has a high pressure, that means it has a low volume. Okay, so if you always think high pressure, that means I have a low volume, or high volume means low pressure. And if I look at my graph right now, it can tell me when the pressure is high and when the pressure is low. So let's look at time y. So time y is in this case here, and time y says that in my aorta and in my left ventricle, my pressure is really high. So I have high pressure at time y. So I'm going to say high pressure. And that means I have low volume. Okay? And if I have low volume in my left ventricle here, that means I'm in the process of squeezing all the blood out. So I'm in the process of squeezing all the blood out. It's not full, it has the smallest volume possible because I'm pushing all the blood out. And if I'm pushing all the blood out, I'm pushing all the blood out. The left ventricle is pushing all the blood out into the valve here, in this valve here. Okay? And it's not pushing all the blood out in the aorta because we know the blood goes from the aorta to the ventricle. Okay? And then the blood is pushed out through the second valve here. Okay? So when I have high pressure, low volume, that means I'm squeezing my ventricle. I'm trying to push all the blood out to go away from me, away from the ventricle. So I know that if this valve is open, that means this valve must be closed because the valves are open only one at a time. So both of these valves can't be open because then that would cause blood to flow in all directions and that would be completely wrong. Okay, so I know that at time y, this valve here is closed because I'm pushing all the blood into the other valve. So at time y it's closed, so I can go ahead, cancel out A, cancel out C. That's pretty good. I'm left with B or D. Now let's look at time X. So again, I'm going to draw the heart again, here and here. Ooh, that looks really bad, but it's okay. Okay, and at time X, I have low pressure and high volume. And what does that mean for my ventricle? That means for my ventricle that it's going to be filled to the maximum with blood. So it has high volume, low pressure. That means my ventricle is filling up with blood. And where is this ventricle getting filled up with blood from? It's getting filled up with blood from the atrium. Because the atrium is sending all the blood into the ventricle. And that means if it's sending all the blood into the ventricle like this, like this, like this, like this, that means that that valve has to be open. So that valve is open in between the ventricle and the atrium. So at time X, I can say that that valve is open. So that's why my answer is B and not D. Did that make sense for everyone? I think Luca said that it's wrong because it's on the right-hand side. Yes, it is wrong. I made that mistake, but it doesn't really matter with the explanation of the question. Yeah, thank you so much. Yeah, user said doesn't matter. Yeah, thank you. Yeah, so it shouldn't affect the explanation of the question. It shouldn't affect your understanding of the question, because it's not related. How do you... is that good for everyone? Okay, so I would suggest drawing it out if you're having difficulties with these questions, especially with these graphs. Okay, perfect. I'm going to move on, if that's okay for everyone. So question 28, almost there. 2 questions left. And then, okay, so question 28 says, which sequence represents the reaction leading to the formation of a blood clot? Blood clot. Blood clot, and then it's the reaction leading to that. Give you a couple of seconds to answer this question. And then, okay, user 5176 said D. Okay. RB3, who's been with us since the beginning, said D. Okay. Lena says D. Okay, so the answer is actually D. So all of you who said D, good job. I'm going to go ahead and explain why that is. Regarding to the steps, I would say you'd have to memorize that. So memorize that prothrombin actually goes to thrombin. When you form a blood clot, prothrombin goes to thrombin. Okay, and that is correct. And all of my answers actually do show that, so that didn't help me much. But one thing you have to know is that when you're thinking of the blood clot formation, you have to think that prothrombin turning into thrombin, thrombin actually initiates the reaction of fibrinogen turning into fibrin. And the way A and B present these options, it's as if they're saying that prothrombin turns into thrombin, which is correct. And then they're saying thrombin turns into fibrin. And that's incorrect because those molecules don't turn into each other. They don't change to become another molecule. What they do is they initiate the conversion of fibrinogen to fibrin. So that's why this like drawing of the line to these lines here, this to this to this, is incorrect because they don't turn into the other molecule. They catalyze or initiate or start that reaction. So it has to be C or D. So I'm already crossing out A and B because that doesn't make sense. Now I have to look at my answer, and from knowing or memorizing or understanding the process of the formation of the blood clot, I know that it's actually fibrinogen that turns into fibrin. So it cannot be C because it's the other way around. Did that make sense for everyone? Hopefully that made sense. Yeah, so RB3 says thrombin catalyzes the formation of insoluble, fibrinogen to fibrin. Exactly, so it catalyzes. Perfect. Why is it not B? Okay, it's not B because the way B represents the blood clot formation, it's saying that prothrombin turns into thrombin, okay? And then it's saying thrombin turns into fibrinogen. And then it's saying fibrinogen turns into fibrin. And what you have to remember is that thrombin does not actually turn into fibrinogen. It doesn't become that molecule. Fibrinogen is already there in the blood, and this thrombin just catalyzes or initiates or starts that reaction of fibrinogen turning into fibrin. That's why you have to look at it in this form of a diagram. Does that make sense? I think Sama asked that question. Does that make sense? Okay, okay, perfect. Okay, question 29, almost there. Question 29 states, the diagram represents transmission across a cholinergic synapse, where a neonicotinoid pesticide acts to prevent synaptic transmission. So I have my 2 neurons, and this is my synapse. I'm going to give you a couple of seconds to review this, and then I'm going to go ahead and answer. Okay, I'm getting a lot of D's in the chat. D, 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 D. D. Perfect. I think everyone's getting this really well. So the answer is D. That is correct. I'm going to go ahead and explain why. Okay, so this is 2 neurons and this is a synapse. And I'm going to first explain what usually happens. This is topic 6, by the way. What usually happens between a synapse, and then I'm going to go ahead and explain what this pesticide does, what it does to prevent. Okay? Someone said y'all we ate. Yeah, you did eat. Okay, so what usually happens is, so electrical impulses pass through the neuron, and then it comes to the end here, and that causes these vesicles of neurotransmitters to release into the synapse, and then these neurotransmitters diffuse in the synapse and come here and bind on this receptor, and once they bind on this receptor, this causes an electrical impulse to be passed on. So this just explains how electrical impulses are passed on. Now, what do we do with these neurotransmitters when we're done? These neurotransmitters usually they're binded, and then they usually just pop off of the receptor site, and they come to an enzyme on the side here, and this enzyme takes them and breaks them apart, spits them out. Okay, and then they usually travel, and then they get reformed in those vesicles, and then the whole process happens again. Okay, that's what happens usually. Okay, but now what happens when we have this pesticides? And these pesticides essentially kill or make those insects not be able to move. So that causes paralysis, basically. Okay, and so how does this happen? And this happens by preventing synaptic transmission. So and pesticide molecule comes here. So it comes here. And what it does is it pretends it's a neurotransmitter, and it comes and binds on the receptor sites. So all of these molecules comes and binds on these receptor sites, okay? And what happens is that they irreversibly bind. So they bind and they never come off. No, sorry, that's incorrect. They bind, and basically what happens, some of them irreversibly, so some of them do bind and permanently stay there, but some of them bind off, so they come off as well. And what happens is, when they try to come to this enzyme cannot break them up. So what happens is they just stay there. And what happens if they just stay there? That means they keep binding again. So I'm binding, I'm trying to get broken off, I cannot be broken off, so I keep binding again. I keep binding again, and I keep binding again. And it's this process of overstimulation, or this overstimulation of neurons. So it keeps passing an electrical signal, and that causes my insect to die or to get paralyzed. Does that make sense for everyone? So it's because these pesticides, they pretend they're neurotransmitters, they bind here, and then they can't break up, and that's why the option is D. Yes. Okay, perfect. Okay, last question of the day. Very proud of all of you who have made it so far. Had some very, very loyal people in the chat. Okay, so question 30 says, for what reason are daily FSH injections given during IVF treatment? So maybe someone can give us the definition of IVF treatment in the chat. That is one thing, or the process. So someone can do that in the chat. Can also explain it, but just for practice, what do you guys think? The last question is always FSH related. That's actually very accurate. Chris, it's always like pregnancy or FSH or sperm or eggs. Yeah, IVF is in vitro fertilization. Exactly, Macy. When an egg is removed and fertilized in the lab. Perfect. And that usually is used for people who cannot have children, so they have their eggs taken out and then they're fertilized in the lab and then replaced back into the mother. B. Perfect. Everyone's getting this last question correctly, which is a good note to end on. So B is correct, to induce the ovary to produce more eggs than normal. So what does FSH? FSH is a follicle stimulating hormone. And what it does, it stimulates your egg to produce. So it stimulates egg production. It causes an egg to be produced. Now, why do we have extra or daily FSH injections given during IVF treatment? Is because we want more eggs to be produced. And the way I remember it is just remembering the full name rather than FSH. I remember that it's a follicle stimulating, and so it's stimulating the follicle for the egg. And then I know that B makes sense because it's saying more production of eggs to produce more eggs than normal, which is perfect. And we do need that in IVF treatment because you have to collect as many eggs as possible in case one of them fails. Yes, user said it's responsible for egg production in the menstrual cycle. Yeah, perfect, exactly. So that's why it's B. Let's look at option A. To suppress the natural menstrual cycle, that's incorrect. To prepare the lining of the ovary for embryo transfer, no. To prevent the development of multiple embryos, no, it's actually has nothing to do with embryos. It's multiple eggs. Okay, that was the end of the session. If anyone has any more questions or wants me to review anything, just let me know. Or if you have any questions about studying in general. Damn, that took up 6 pages in my book. Are there more sessions like this? Well, we currently don't have any schedule, but if you guys would like, it's depending on demand. What is the best way to study for bio? Okay, what is the best way to study for bio? Well, for me personally, how I studied for bio is I found a way to learn the content, first of all. So learning everything or reviewing everything because I didn't want to retake notes. So for example, for me, I actually joined the crash course that I'm now company like working for. And I joined the crash course and I was able to learn everything. And because I was able to revise the whole syllabus, I didn't have to like take time writing notes. If you can't join the crash course, maybe you can watch like YouTube videos on all the content and stuff. And then I think the key thing to my revision, specifically for the IB exams, is how many past papers I did and how many past paper questions I did. And that's kind of why we had the session. Hi, I've sent the QR code. Students can choose Christian iPad. Okay, perfect. I'm going to share a QR code. Give me 1 second. Just for feedback, and that actually helps you demand. Are you by any chance Arabic? Yeah, I am. Yeah, and then with this QR code, if you answer this QR code and are able to give us honest feedback and like demand more sessions, of course, we'll be happy to do so. So I'm just gonna find some way to show you this. How do I? Can you please have more SSS? Okay, so I'll send in a QR code right now, and if you guys give us feedback, how will we know? How will we know? I was just like following and keeping updated with the account. Usually when we have a live session, we put it in the bio and it's put in as a live event. Is there going to be a recording of this somewhere? Yes, it will be on YouTube. So I'm just gonna share the QR code real quickly. I'm having issues sharing the QR code. Thank you so much, Air. HL stuff would be great. We want more bio SL. Okay. What I'm gonna do is I apologize, guys. This is our first time doing a live stream, so still getting used to the hang of it. So I'm just gonna. Is there a way to? Okay, anyone, can you do some past questions be repeated? Yeah, the best way to revise is past paper. I think so. Yeah. So I completely stopped my train of thinking. But yeah, so I would find some way to review all the content very fast. And I would say stop yourself from just taking a bunch of notes. And once I've found a way to review the content very fast, YouTube videos, the crash course or whatever, then I would just push, and cram past papers. And once I've crammed those past papers, I would see what questions I got wrong and what topics I need to improve on. And after doing that, here I've solved the issue. After doing that, I would go back and review which things I had trouble working on. Okay, now. Oh, thank you so much, Daria. That is very helpful. I'm gonna try again to do it. And if I can't, then I will do exactly what you said. That was a very helpful tip. Do you think questions from past papers might be reused? I wouldn't say exactly reused, but like rephrased, yes. I've seen that a lot, especially in chemistry, for example. If you take chemistry, I've seen that done a lot. they're not word for word like reused, but it will be the same, basically the same exact thinking, the same type of question. And also, as I don't remember, I don't know if you guys were here when we were talking about it, but someone said that they always end with like a pregnancy or like a hormone question about like FSH. And that is true. They usually have a pattern of what questions they put in first and what questions they put at the end. Do you guys answer emails? If we have doubts about the crash course question, of course, just send us an email. We also have new option topics. So if you're interested in cramming your options, because for example, my teacher never taught me my option, so I had to learn it all by myself. That's why we introduced the option topic. I'm not really sure how to. Okay, I've got it. I've got it. Okay, add image. Okay, here's the QR code. Yay! Thank you for being patient with me. Okay, is this big enough for everyone to see? Did you get a 7 in biology? Yes, I did. What option topic? What is the option topic? So like option A, option D, like you know those option topics? How much can your grade be improved and how to perform in paper 3? By how well you perform in paper 3 compared to paper 3. So you're maybe you're asking like if I do really well in paper 3 and then paper 1 or 2 compared to paper 1 and 2. So I would say that I got a lot of advice from my teachers to not study for paper 3 and that paper 3 wasn't important. But actually, do not listen to that at all. That is the worst advice someone can give you. I would say definitely work on paper 3 because the option topic or paper 3 is a much shorter syllabus. So you can work on perfecting that much faster than you can do your whole syllabus. And your paper 3 is actually worth 20%. And that's the same worth of your paper 1. And I remember walking out of my paper 1 and I was really stressed that I did so bad, but I had my paper 3 right after my paper 1 and one of my friends told me, "Okay, calm down, don't stress. You still have paper 3 to do, and if you perfect paper 3, it doesn't matter what happened on paper 1 because that can still carry your grade." So, I would say prioritize all, make sure you distribute your learning and everything. Um, and you know paper 3 well. But if you know that you're gonna ace paper 1 and ace paper 2, then maybe you don't have to put as much effort in paper 3. But I wasn't so confident. Yeah, exactly. So Simon said, or Simon, sorry, said, my teacher said the opposite, that we should put most effort into paper 3 in case paper 1 and paper 2 didn't work out. Yeah. So maybe not put the most effort, but put as equal effort or put as much energy. Is the crash course similar to this? So the crash course, actually, you learn the content, or you review a content with our tutor, and then she'll give you content, and then she'll give you past paper questions. So she handpicks the past paper questions related to that topic, and then you work out the questions with her in the topics, and then um, she'll show you the mark schemes. You'll do both paper 1 and paper 2, and then she'll go ahead and explain something else. So then she'll explain the next topic, and then it's like a day. So the crash course is 5 days, and um, yeah. So throughout the whole day, you just learn a bunch of topics. Are you the teacher for the crash course? No, I'm not. If you go on our website, you will actually see the teacher. Thank you so much for this course. It was super helpful. Of course, we are very happy to help. Where can I join paper 3 sessions? Yeah, I think they will let you know in the chat. But on our website, everything is on our website. So if you're ever confused, just be. Are you going to be the teacher? I am going to teach the option topics. But for the crash course, I wasn't too confident in teaching the crash courses, so I wasn't open to doing it. But if you guys would like me to teach part of the crash course, I would be happy to do so. Thank you so much, Leona. I wish you the best in your studying. If anyone doesn't have any more questions, I'm going to end the live, if that's okay with everyone. All right. Okay. All right. Thank you so much, everyone, for joining. I hope today's session was helpful. Please don't forget to scan the QR code and give us feedback. So request chemistry, HL or SL content, and we'll be happy to do more of these if you guys want some. Join our crash course, join our option Ds, Use all the resources on our site. If you have any more questions, feel free to message us on TikTok or email us. We are more than happy to help. All right. Bye, guys. Okay, I think I'm still live on YouTube, so I'm just going to stop streaming. For everyone on YouTube, thank you so much for joining.